Neil, would you like to start?
Sure. Well, thank you, Jim and Andrew, for just I guess asking me to do this. I'm I'm pretty excited because it brings me back to like my PhD days. I actually have slides from there in this presentation, and I'll be making some comments about my style back then. But anyway, um, so I really want this to be informal. Just if you have any questions, just interrupt me. The chat is going to be a little difficult, so it'd be better if you just kind of spoke up. And so so this is about RIPSeq, and this is my sort of, I guess,
Neil,
day late.
Just a real quick note: if you guys do want to, if you don't want to interrupt Neil, and type something in the chat, I'm happy to interrupt you, Neil. So I can let you know. So
yeah, that'd be great. I think this is really like for you all to get as much as you want out of at least my thoughts on this process. So yeah, so here's my sort of day late homage to St. Patrick's Day of seeking. And if you RIPSeq, you shall find RNA, hopefully. And I will try to give you tips on like making that better than hopefully. So just an overview of how I have this structured, a little bit of background on post-transcriptional regulation and RNA binding proteins. Since this is a cancer center event, I figured I'd give a little background on why study RNA binding proteins in the context of cancer. Then we'll talk a little bit about like the variety of ways of thinking about how to identify a target of an RNA binding protein. And then we're only going to talk about like a little sliver of that. And in that, I'm going to go through sort of experimental considerations, analysis, and interpretation. So RNA and gene expression is quite involved. It's a little bit more than just DNA, mRNA, protein. There's a whole slew of individual steps, and they are actually not sort of randomly happening. It is a highly coordinated set of events. So if we think about an RNA being transcribed, it gets capped, right? And then this intron starts getting spliced out while it's still being transcribed. The RNA gets cleaved, polyadenylated, exported into the cytoplasm where its localization, decay, and translation are all regulated. Oops. So as you can kind of see, in all of these cases, all of these RNAs are decorated with proteins, and these proteins will bind to specific elements in these RNAs and dictate their fate with respect to any of these processes. I'm going to give you like a quick overview of how to think about this from like a regulatory logic viewpoint. So this model of sort of RNA regulons is a way of thinking about the coordination of post-transcriptional regulation, and it was posited by my PhD advisor and a while back. And I think there's some really interesting specific points that will help you think about what you might expect to see when you kind of study an RNA binding protein. So RNA binding proteins bind to multiple RNA targets, and oftentimes they encode functionally related proteins. And so, one way you can think about that is that that's a trans-specific binding, right? There's a combinatorial regulation there. And so if we were to just look in this picture, right? So we have a bunch of genes 1 through N, they get made into RNAs. And then, these boxes kind of show regulatory elements. And, so the yellow ones are bound by this sort of yellow RNA binding protein, binds to multiple RNAs. And oftentimes these RNAs encode functionally related proteins. So that's kind of an interesting way to coordinate gene regulation for a specific purpose. The other interesting thing is a single RNA is often bound by multiple RNA binding proteins. And so you can kind of see that, not in these pictures per se, but you see how there's regulatory elements within these multiple regulatory elements within an RNA, right? So it's bound by multiple RNA binding proteins. And sort of more recent work has shown that the ensemble of RNA binding proteins bound to our RNA will play a large role in what happens to its decay, translation, processing, so on. And the last is that that this sort of regulatory environment, these interactions and the regulation itself, is very stimulus responsive. And that's something that is not as well characterized. And I'll tell you a little bit about that. So when we think about, I kind of gave you a view of, how RNA binding proteins control different aspects of gene regulation. So these are sort of very general examples of what can go wrong. So you could have, aberrant expression or localization of a given RNA binding protein. And so then it starts, having different substrates than it typically would. That can result in deregulation of processing or, stability, translation, localization. You could have, these can have really important impacts on these different processes, and then on specific sort of pathways that would result in different phenotypes. Now, I'll show you sort of a different way of thinking about this here, right? So here are RNA binding proteins that have been implicated in specific cancer-related traits. And as you can see, this list is, there's actually, proteins that are in multiple parts of this list affecting different aspects of these different traits. But sort of suffice to say, there's actually quite a lot of evidence supporting how that RNA binding proteins can be really important in the context of cancer. None of this is really specific to any cancer. I'm just trying to give an overview. So it kind of begs the question, by knowing what some of the targets are, you could start really understanding what's going wrong in gene expression in these contexts. So that's a little bit about why you should care, I guess. So how do we actually find these targets? And this is a figure from a review paper, and basically this is just like a little piece of an RNA, okay? And they're depicting, in this case, these blue proteins are double-stranded RNA binding proteins, the green are single-stranded, and then this is what we're calling an indirect RNA binding protein, right? So it's not directly bound to the RNA, but it's part of this complex. And so there are a few ways here. So this UV, this is a different flavor of UV. The 4SU is not super important here. And formaldehyde, these are all ways in which you actually cross-link the protein to the RNA, as you see in these like red X's. What's interesting, so UV, these are both UV, the PAR and the UV, and that is a zero-length cross-linker. So you only get direct interactions. Whereas formaldehyde has a wider radius of cross-linking, and you actually cross-link some of these sort of indirect proteins, so like to get complexes. So there's also methods where you don't cross-link, right? RIP, and that's what we're going to be talking mostly about today. And in RIP, there's sort of the standard RIP, and then you can even like kind of digest the RNAs to get a little bit better resolution. Again, we're only going to talk about this, right? So depending on the protein that you're studying, you would make kind of different decisions as to, what the appropriate strategy here is. There's also ways in which you can fuse, A-to-I editing domains onto an RNA binding protein and then read out the mutations in the sequence. That's particularly powerful when you have, say like a rare cell type and you're trying to understand what the targets are. You don't have a lot of material. You could drive the expression of that fused RNA binding protein in only the cells you're interested in, and then try to get a readout by RNA-seq. So there's a whole ton of methods. We're really just going to be focusing on this one for today, although I'm going to bring in a little data to give you an idea of how these compare. Okay. So, oh, it's not animated. That's annoying. Oh, well. So just follow my cursor here. So the average RNA, look in humans at least, and mice, looks something like this, right? So you have, 200 base pair, that's 0.2 kb, 5' UTR, a kb coding sequence, and a kb 3' UTR, and a poly A tail, just like in the Zoom link. And if you think about this, right, that's a lot of there's a lot of sequence kind of here and here that you could imagine there could be binding, right? The coding sequence, if it's translated, the ribosome is just kind of like coming through it, and you could also therefore imagine that might that proteins are not going to withstand the ribosome, right? It's a pretty massive machine with lots of helicase activity, and so oftentimes we think about the 3' UTR for binding, but realistically proteins can bind all over. So here's our RNA, right? And now if we kind of look at these are just like a decoration of proteins on there. And if we kind of zoom in here, one of the first considerations we have is like, do we cross-link or not? And then do you do an RNase treatment or not to get to like a binding site level, right? And and then you have to consider whether you are doing a protease treatment to get rid of the protein here so that you can actually kind of sequence this little bit of RNA. So there's lots of things to think about here. What we're really talking about with RIP-seq is really the full length, okay? So you're getting the whole RNA. There are, there are considerations in terms of, lysis buffer that you want to use and where the RNA binding protein is localized. Certain lysis buffers are sort of more for a cytoplasmic, so you don't get a lot of nuclear lysis. So anyway, technical considerations, I'm happy to talk about it some other time. Just keep them in mind. Depends on, again, on the protein you're studying. There are some biases in terms of being able to immunoprecipitate a really long piece of RNA versus a short piece of RNA, right? So precursor RNAs are going to be a little bit harder to IP, and so, but that doesn't mean it's not possible. So this is a paper from the Rio lab, and this is a RIP tiling array seq, not seq, but basically you can see that you get a lot of coverage over introns. So it is, formally possible. You just got to worry a little bit about kind of bias. Okay. So again, this is a matter of resolution, like transcript versus binding site, and sort of location of the RNA binding protein. Okay, so here is a figure kind of describing what this looks like. This is from an older review that I was part of, and apparently I must have been reading a lot of Harry Potter, because I almost have like a Deathly Hallows symbol here, maybe with the antibody, but anyway. So we have these cells, and you lyse the cells, and you get a whole set of RNAs and RNA binding proteins, right? And then you do an IP with antibody specific to your RBP of interest. You also oftentimes do a mock immunoprecipitation with an isotype matched antibody. We'll talk about some of these issues in a bit. And so, if we are getting these red ovals, right? So then we're enriching for those here. The mock will basically sample, just randomly sample, ideally, out of this pool. You also honestly actually want to look at the input as well. So basically just, you want to kind of get a sample from here and see what that looks like. I'll talk about the comparisons in a little bit, kind of which one makes more sense or works better. And then the nice thing here is you have RNAs and proteins now, and you can actually look at both components, either with proteomics or with sequencing, or an array in this case. So these are ribonuclear protein complexes, right? So the RNAs and the proteins are kind of important to think about, but we're going to be really talking about the RNA. And I'll go through a couple of these considerations in the next few slides. So the first thing you kind of want to do is, can you enrich for the protein? This is pretty important, right? So here we're talking about ELAV-L1, which is also known as HUR, and it's a flag tagged version. Okay, so there's both a flag tagged and an endogenous version in this cell. We're going to just focus on the flag lane here. So in the input, you have a flag, right? Here's an IgG1 IP. Here's a supernatant for the mock IP or for the ELAV-L1 IP. You can see there's, relative to the IgG, there's a little bit of a depletion. So you don't necessarily expect that you're going to deplete all of your protein. It's rare that happens, especially given how expensive antibody is. And then with the IP, right, in the IP lanes, you see basically no flag tagged UR here, ELAV-L1, and you clearly see it in the IP lane, right? This is just showing like what's happening with the endogenous. Here's the actual endogenous protein, which is not getting IP'd. So that's because you're using the antibodies against the epitope. Okay, so then you kind of look at enrichment of known targets, and these are all targets of this protein HUR, and this is by qPCR, and you basically see an enrichment. This is normalized to a non-target GAPDH. You see enrichments of beta-actin, TNF, cyclin A2. And in some sense, you actually see a little bit of a higher enrichment in the input versus the IgG, but this is really like, either way, you want to look at this before you kind of commit to the sequencing. Oftentimes you don't know the target, and this is like a dirty trick that you have to be a little bit careful with, but RNA binding proteins are very, very likely to bind their own mRNA. So if you're at a complete loss and you just want to like try something out, that's one thing to do. But you need to be careful, because you could also imagine a scenario where the protein is starting, the RNA of the RNA binding protein is being translated, right? And you get a little bit of the polypeptide coming out and starts to fold, and then that gets recognized by the antibody, and that's why you see the enrichment for the actual mRNA of the RNA binding protein. Anyway, there's ways to get around that, but point is, this is actually a feature of these post-transcriptional networks that's really interesting, but I won't go into that. So,
Neil, can I interrupt you and ask you a question?
Yeah.
Okay, thank you. So I do see this in a lot of paper with these RNA binding proteins, that people will add these tags to purify them. And it also, like, I know that there's not always great antibodies against the RBP itself. Is there evidence, however, that when people add these tags, it affects the ability of the RBPs to, localize subcellularly? Or has there been evidence in the field that any of the activity of the RBP is affected?
Well, so I mean, like, I feel like anything, it's sort of protein specific. There's not a general rule where, like, I don't know, all RBPs are kind of damned to not work once you give it a tag, or the opposite, right? That you're good, right? So, I mean, same thing is you kind of need to check. I think that another consideration is if you start playing around with the stoichiometry, like if there's endogenous and tagged in the same system, then you could start, hitting suboptimal targets, right? You could start getting yourself into a little trouble there. The tag itself, like, honestly, any other protein, you need to do a little bit of due diligence.
Yeah.
But
okay.
So the other thing is for if you're doing cross-linking and you're doing more harsh washes, that's where the epitope can help too, because like flag, for example, that antibody can withstand a stronger wash than some of the antibody antigen interaction can withstand a stronger wash than, say, some of these other antibodies, right?
Thank you.
Yeah.
So Linda had a question in the chat, Neil.
Yeah.
Why do many RBPs bind their own mRNA?
That's a really good question that we don't totally know the answer to. I'm going to give you the hand wavy speculation that it's part of a way to buffer its own expression. And so RNA binding, when you look at large scale compendia of targets of RNA binding proteins, it's not only that they bind their own RNAs, but they bind each other's RNAs. And so I think that it kind of speaks to, like, there's mechanisms to not let things get out of whack too much by having this redundancy built into the system, which is also kind of a pain in the butt for genetics.
We've seen it, but it's kind of reassuring to know that it's normal.
Yeah, I mean, it's so normal that I would even like suggest this as like a one-off, give it a shot type thing.
Hi, Neil. This is Adeli. Kind of going off of Katie's question, I'm curious if you have any idea how N versus C terminus tags could affect the RIP-seq process, or is that depending on, again, like which RBP you're looking at, or?
Yeah. Okay, so I think, like, maybe to try to be more helpful, then you have to check it. You like maybe looking at the domains of the RNA, like the domains within the RNA binding protein, and where they are relative to the N and C termini, right? That could help you to make that decision. There's not like a I mean, I don't know. I prefer to try to tag the N terminus, but I don't have a great reason for that. I mean, realistically, you would have to test it, but staying away from domains, like, I think is a good idea, right? So like one of the proteins we study, like the very, very C terminus, is where it recruits the deadenylase from. And but the first, like, 20, 30 amino acids don't seem to have a known attributed function. And so that's a rationale for saying, okay, well, let's really start with the N terminus rather than mess with the C terminus. I don't know if that helps.
Neil, can I comment?
Mm-hmm.
In some of our receptors that we study that are post-transcriptionally regulated, the C terminus is actually processed prior to reaching the receptor reaching the membrane. And so if you put a clip, not a clip tag, most often the tag never ends up on the membrane. So people, because of the C terminus processing that happens in many proteins, they prefer to put it on the N terminus right before, even if it is a signal sequence containing protein, right after the cleavage site of the signal sequence.
Yeah, that's a great point. I mean, I think there's, biology is interesting enough that it can kind of, mess up your plans in so many ways. But I mean, I'll agree that we usually start with the N terminus as a starting point.
So Neil, maybe extending a little bit on this topic, what's the sequence homology between the RBPs? Are they just completely diverse, or are there some commonalities between them?
Yeah. Okay. So within specific families, right? So like, okay, let's take ELIVL1. So there's ELIVL 1 through 4, and they each have 3 RNA recognition motifs, and within the RNA recognition motifs, you're talking like 90% similarity. But then outside of those regions, it's pretty divergent. I would say like 50% similarity. And the RRMs from human to mouse are very, very similar. Now, across RNA binding proteins, it varies. It really depends who you're comparing. There are like a number of, so it gets a little tricky because recently there's a number of proteins that are found to bind to RNA through non-canonical means without a defined RNA binding domain either. So once you open it up to those, then it's really, really out there, right? But there are a subset of like specific domains that are known to be RNA binding, including like RRMs, zinc finger. There's zinc fingers that bind RNA. Oh, yeah. If you're studying a zinc finger protein, make sure you don't put EDTA in the buffer, so you don't chelate the zinc and not let it bind the RNA. So anyway, yeah, I don't know. That get at what you're asking?
Yeah, I'm just curious on, maybe this is a silly question or a bigger question, but I'm just curious on what is the kind of the landscape of RBPs in general, and like how many proteins are we talking about? Do we know?
Okay, conservative estimates, I would put it at 1,500. And that estimate has only grown with experimentation, but where now we're talking about things that bind RNA, but without a clear domain. And even that 1,500, not all of those have a clear domain, but they have multiple lines of evidence. They vary from, this will be useful for the next slide, you have things that bind to ribosomal RNA, to SNO RNAs, snRNAs, mRNAs. I mean, there is some crossover, right? Like, it's not like, they know the type of RNA they're binding. But that's one way to think about them being different. Another way is the number of targets they have and their specificity. There's some that are exquisitely specific and have like, bind a, really, specific 8 nucleotide stretch. There's others that bind like GU and just like, bind everywhere, right? That's how those are the ways that I think about the breadth of RNA binding proteins.
Thanks.
Yep. Okay, so then you need to think about, what what is the RNA that you think the protein binds, right? So here is like a table of all the different RNAs that we know, and it's ordered by the sort of mass in terms of the total RNA, percent of total RNA by mass, right? But then what's really important is thinking about the length, right? So some of these are well over a kb or more. Some are quite small, like tRNAs and microRNAs, right? So you really have to think about the library preparation that you use for this, right? And so one tip I'll sort of mention is that you can do some of these extractions with columns, but I would also recommend like thinking about just using glycogen as a carrier to make sure you get as much RNA as you can, because you don't get a lot of RNA out of this. Because if you think about it, especially if we're talking about something that doesn't bind all RNAs, right? So you don't get a lot of RNA, and it's at the level where I don't even trust an antidrop. So so just be careful with this. So if we think about the different RNAs in the cell more graphically, you have to consider which RNAs you're interested in, and that means either doing things like poly-A selection, depletion of ribosomal RNA, or size selection, right? These are all like, and these are not mutually exclusive either. Typically they are, but they don't have to be. And so if you're looking at kind of these populations of longer RNAs, usually you get the RNA, you fragment it, and then you do random priming and you get your library, and that's across the whole transcript, right? If we're talking about sort of this is more like a microRNA library preparation. So you need to really consider, so here you get like a pretty small fragment and you kind of stick adapters, you ligate adapters onto the 3' and 5' end. You got to be careful of what these ends look like in terms of whether they have a phosphate or not, and a whole variety of issues, right? But microRNAs are ready to go into a lot of these. Other RNAs may not be. So anyway, this is sort of like the consideration here, the size of the RNA. Okay, so the ultimate goal here is to kind of increase signal to the background. And so, ways of like really getting at the background are like doing mock IPs, like I said, with an isotype matched antibody. Also getting the input RNA. The thing that's sort of nice about these is that you're actually dealing with the same pool of RNAs, right? That you're pulling out of for the positive IP. Another good control, but a little different, right, is when you don't have the antigen you're looking for, right? So you do a knockout of the RNA binding protein. But now this input, the pool is different, right? Like the transcriptome is going to be different. And so you have to then think about taking that into account in terms of saying like, okay, we have enrichment or no enrichment, right? So anyway, things to consider. I've played around before with spike-in mRNAs, like putting in ERCCs to help with normalization. It's like one of those things where like when you don't do it, that's like going to be the time that you need it. But I don't know. I for a number of RNA binding proteins, like kind of typical mRNA binding proteins, it doesn't seem to have been necessary. But it's something to consider. And then think about the beads, right? So like different beads have different properties, so that will affect the incubation time and how you couple the antibodies. And then of course, like the standard chart of like, the type of antibody and the type of bead, right? Protein A, protein G. Okay, so let's get to the fun part here. So this is what the data actually look like. Okay, so this is again, so that Western and qPCR that I showed earlier, this is the sequencing data from that. This is beta actin, which is a known target of this protein, HUR. And so this here is the locus. And so the thick lines are the exons. These are the introns, exon, intron, so on and so forth. These libraries were actually ribosomal RNA depleted. So this wasn't a poly A enrichment. This was a rRNA depletion. And so in the input, these are all normalized. So like the depth, it's depth normalized so that we can really compare the height of each of these, of the coverage here, right? So what you can see is in our RNA binding protein IP, we see a lot of reads, and it's really over the exons, right? So that sort of indicates that it's the mature RNA that's being enriched. And you don't see much in the input or the IgG. You do see stuff though. So like, this is not, it's an enrichment. It is not by any means a, like, I actually think sometimes this background is helpful to think about this in a more quantitative way. And I'll explain that in a minute. So you basically make comparisons between, either the HUR and the IgG or the HUR and the input. I'm not showing this to you right now, but like, actually we didn't see much difference when we compared the two, like whether you compared it to the IgG or the input. So I think if you're trying to like cut costs, I would say just do the input, because the input's a more valuable sample anyway for other comparisons. Okay, so now we're going to shift into the analysis of the data. And I didn't realize this. So I just like kind of just Googled around, and there is this like post on BioStars, which is like, a place where people ask questions like RIP-seq. And there's a whole host of information out there. And I think there's a lot of issues with RIP-seq analysis. And I think it's actually being overcomplicated in a lot of these forums. But the caveat is what I'm talking about is RIP-seq, where you have the full transcript and there's no digestion. In that case, peak callers are not appropriate because you're enriching a whole transcript that has a structure of its own. And so, there's a lot of like, using ChIP-seq methods and things of that nature. And you should really stay away from that unless you are doing a digestion or you have some other reason to do that. If you don't have a well-annotated genome, then that's a separate thing. But, yeah. Okay. Okay. And yeah, so like I said, some of these considerations are going to be dependent on whether you do like a small RNA or a normal like long RNA. But I would really recommend these sort of two, oh wait, we'll talk about this. So splice aware or transcript-based alignment and quantification. And then we'll talk a little bit about detecting enrichment of the transcripts and motif analysis, which is, I think, common things that people want to do. So there are about three software that are kind of RIP specific. The problem with these are that so they're not really actively maintained, and with some of them they are peak detecting methods, and you don't really need that, and it can actually cause some issues if you're again looking at sort of full-length RNAs. And so I would really kind of recommend to just treat it like RNA-seq. And using DE-seq2, that will work fine. I'm going to tell you a little bit more about how to deal with it, but realistically, you can use some of your typical RNA-seq pipelines for this. Okay, so remember the things that I just showed you one gene, right? And this is, sorry, one RNA, and this RNA is enriched, right? So what do we kind of really want to look at here? So what we really are doing is we're making a quantitative comparison here between, the RBP IP and the MOC or the input. And so what I would really suggest here is, especially for like human, mouse, right? Most model organisms, is that you use salmon and you just do quantification and kind of like pseudo alignment. If you suspect your protein binds to precursor, like pre-mRNAs, right? There, you can still actually use salmon, and there's ways to kind of spike in the whole locus, and that works actually pretty well.
So Neil, I was going to ask about the de novo type of discovery versus, the quantification of known transcripts type of issue. So how do you do that? And is there, I guess I don't know, can you use it for like de novo transcript identification?
You could. So I mean, I've never tried this, but I don't see why you maybe couldn't like stick it into some of the transcriptome assembly approaches, right? And is that what you're asking, right? Like to basically like reconstruct the transcripts that you're getting?
Correct. Yeah. So assuming it's, you pick up the known stuff, right? The common transcripts that are annotated. But what about the new, are there new novel things that you could pick up with this technique that I'm just not aware of?
Yeah, I think you could. I personally have not tried it. I think it could be really interesting, especially in cases where, like there's proteins that could be binding to RNAs that are like improperly processed, right? Like that their job is to like stop that, and that transcript might not be annotated, and it might be part of like a normal like gene that we know, right? And so in that case, something like what you're suggesting could be really valuable. There also are ways of doing like kind of differential expression across bins in a genome, which is almost a little bit like a peak calling-ish exercise, but more just treating it like RNA-seq. That could be useful too. I think that was from Jeff Leaks. No, I can't remember. Anyway.
Linda had another question in the chat.
Yeah.
I guess it's to either me or Neil, and both of us. How would you curate those, assuming the novel transcripts, from those that do not map?
Well, okay. So I think there's a couple things in there, right? So like with some of the transcriptome assemblies, you still usually use the genome sequence to guide the assembly. So so that's like the starting point, because anything without the genome, I think, gets really scary with short reads. But in that case, when you do those assemblies, you get, it kind of annotates it for you. Like it at least says like what kind of genes it overlaps with, if it shares, splices. And so that, you can get information just from that process as to, what that, where that transcript might be associated with. Yeah, I don't, Linda, if you're talking about some of the
one
viral.
of the things I'm really curious about is both for viral and host. There are RNAs that we've found in other ways that are 3' to tRNAs, and they actually get transcribed, but they don't map because supposedly it stops, but it doesn't always stop. So we're interested in knowing more about these, but normally, because the RNA supposedly ends before these. So if you were to map them and the software didn't automatically throw them out, they would be adjacent to a tRNA, but a lot of the software automatically tosses them.
So in your case, if you know them, then I would just add them to the database of transcripts that you want to kind of align to.
We know them for the viral ones, and we have done that. But we're interested in whether that's a conserved thing and whether the cellular genes have some examples of that as well.
Yeah. So I think you're right. It's going to take a specialized analysis in that case. I think I don't think you're going to get any out of the box for dealing with that in the human genome. But it sounds like you have a pretty specific question you're asking, and it would be pretty reasonable to set that up as a like to look for those particular cases. So I'm happy to talk to you more about that, like whenever.
Yeah, can I ask you a quick question while we're here too?
Yeah.
So in my case, for example, as you probably know, we would just be interested in just what transcripts are being bound by a particular RBP. And I guess to kind of get away from just, the most enriched hits and maybe get a full picture, how deep do you recommend sequencing?
I think if you're doing this on a gene level, like gene level targeting, I think the same. And we're talking about mRNAs. I think that you the same numbers as for RNA-seq work, right? So like, 10 million per sample will get you pretty decent like answers for, I don't know, 10,000 genes, right? Like you would be pretty high confident. If you want isoforms, you gotta go deeper. Like if you want transcripts, you really gotta step it up and get into the many tens of millions, yeah.
Okay, thank you, that's helpful.
Yeah. Okay, so I want to show you what happens when we compare these two to each other, but look at the distribution for all genes. And this doesn't always happen, but when it does, it's really cool because it tells you, it's a really nice way of comparing sort of targets, right? So this is from array data, and I'll show you later that it pans out for sequencing data too. But this is a distribution of the t-scores of the RBP IP versus the mock. So that's basically just like a variance moderated mean, right? And so over here are things that are really high and consistently high in the RBP IP. And over here are things that are low and consistently low in the RBP IP, like higher in the mock IP. So you could probably already see that there's sort of like a bimodal distribution here. And this is cool because it's kind of telling you like, okay, well, this is who we're interested in. And so like the simple way is like, this is background, this is enriched. Again, remember, our goal is to always try to figure out a good way to separate, signal and background. And so what you can actually do is fit like, you could think of this as a mixture of two populations, and you can fit two Gaussian curves to this data. It's called a Gaussian mixture model and was first used to figure out carapace sizes in 1894 by a really racist person, but basically found differences in species of crabs. It was pretty interesting. Anyway, I will not get distracted by that. So the thing that you do here is that basically at any point on this line, you can ask, okay, what's the probability that I'm in this population versus this population? So that's kind of nice because you get this sort of essentially like a log odds ratio of whether you're bound or not. The other thing is we can have a way to sort of make a cutoff and say, okay, here are our targets. So you can go either way, kind of a discrete approach, say target, not target, or the sort of a probability of binding approach. And so here's an example for in RIP-seq where this also kind of played out. This is with the SM protein. That's from the Matera lab, and they were able to really like kind of nicely identify a subset of RNAs. So this is sort of like a post hoc step, like after you've kind of done some sort of comparison of the enrichment. So the x-axis in this case is, just some version of enrichment. Here it's just a log fold change. And so what's kind of nice about this, I was alluding to this, right, is that you can basically, get an idea of like yes or no in terms of what's a target. You can have like a continuous measurement that you can use and plug into different approaches. I'll show you that in a second. And then these can feed into the types of things you want to do, right? Like, okay, do these mRNAs encode common functionally related proteins, or what are the motifs? We've actually taken these like measurements into the CMAP database and then found molecules that like apparently affected QR function, which was kind of neat. I think we got lucky though, but I don't. Anyway, I mean, who knows? And then we can also compare the changes, the strength of binding, and how that changes either across like a condition or across different RNA binding proteins. And I think that's also sort of valuable to think about this in a little bit more of a quantitative manner, not just bound or unbound, but strongly bound or weakly bound. So one of the very first things you might try to do is identify the regulatory elements, right? And so basically what you're going to want to do is for whatever RNAs you're interested, you need to collect the, we're going to just talk about 3' UTRs right now. So you collect the 3' UTR sequences, but really whatever sequence you want. You could use the whole RNA, but there's some, with the coding sequence, you have to be careful because there's codons and you got to be careful of mixing signals there. But there's sort of two approaches that I want to highlight. So one thing you could do is you could just take like, the set of RNAs, look at their 3' UTRs. What you've probably seen like analysis with MEME, which is a kind of classic software to do this. I like a different variant of this, which is Motif RG, that does a similar kind of thing, but actually has this really cool sort of step where it will sort of like mutate the motif and then see if that increases or decreases the enrichment. And then basically it's a greedy search to figure out what might be the best motif that is characterized in your set of RNAs. Another sort of greedy approach where you actually use the whole data set essentially. So this is where having that continuous variable is kind of nice. You can just look at enrichment of motifs in 3' UTRs that are ranked by the evidence. In this case, this is like binding evidence, right? So if there's some of you that are familiar with GSEA, this is like GSEA for motifs, essentially. Same kind of approach, but there's this cool like evolution process, just like there is here, to kind of optimize for the most likely motif. So anyway, I would recommend those as software. One sort of so shifting gears to some of the interpretation, I think this is sort of an important thing to consider. So there's a lot of single-stranded RNA binding proteins. I'm showing you 2 RNAs that have binding sites for 2 proteins, right? But in one case, you know, this same sort of region where this purple element is now embedded in a secondary structure, okay? And so you might imagine that this precludes binding to this region, and this turns out to be the case. So this was a study from Quade Morris's lab, where they basically tried to classify whether a transcript was bound or not by using 2 measurements. One, which is this number of TS, so is the number of like motif matches in that RNA. And the other is the number of accessible motif matches, right? Accessible target sites. That's what TS is. And so in each case, or not in each case, but in the cases with asterisks, you really see that the classification of target versus not works a lot better when you take into account the local secondary structure. And so so this is not surprising, but I think it's pretty interesting, and actually shows you other things you might be able to do with this type of data. Okay, so one thing I'll point out is that this is a data set from my PhD where there's 3 time points where these Jurkat cells were stimulated with it was like a T cell activation, essentially, if we can call Jurkats T cells. And we basically, you know, found, you know, 600, 800, and 900 targets at different time points. So there's a total of 1,200 targets. Only 400 of them were bound through all time points. So this is basically just saying that the majority of these targets were not constitutively bound, and there are these changes in binding. And so, you know, this is something to consider depending on like the biological system you're studying, that you could really see remodeling of these RNPs. Since we could kind of have these like quantitative measurements of association, you could also ask some interesting questions, right? So you could say, okay, so the LOD is the log odds ratio of binding. And this is a correlation comparison where, you know, if we look at the 0-hour LOD score, so the likelihood of binding to HUR, and the expression level, right? You see that there's very low correlation, which is, in some sense, it's good because it's basically saying that you're not binding RNAs just because they're abundant. There's specificity to the enrichment. And that's true for sort of all the time points. Now, if you compare the correlation between total RNA, like essentially the inputs, so there's quite a lot of changes in the over the time course, right? So the correlation is relative, you know, 0.8 or so, right? So there's a decent amount of RNAs that have changed, but it's even more dramatic for the HUR association. So all of this is just to say that this is a really dynamic regulatory environment, and so it's worth really considering.
So is the remodeling specific or random? Like something dictates.
Sorry.
Like you said, 400 targets are constitutively bound by the RNA BP or RBP, but when it comes off of the RNA, is that regulated itself, or it's just a temporal event?
So, okay, in this case, this protein actually shuttles. So in the resting cells, it's like 90% nuclear. And then when the cells are activated, then it shuttles to the outer nuclear envelope, rough ER actually, and a little bit to the cytoplasm. So it redistributes where it is. And there's changes in expression too, right? I think the combination of those are basically what's leading to the changes in binding.
Got it.
All right. Okay, so one thing that I have to bring up is that with this technique, there's no cross-linking. And so you have to think about reassociation of the RNAs after lysis, right? So what you could be getting like targets of your protein, but they might not be like the physiological targets. And so this is something that can be really problematic or in some cases not problematic at all, right? So the thing to really think about is the type of RNP that you're analyzing and its sort of biochemical properties, right? So the lysis procedure will be relevant here. So SNRPs versus like mRNPs they're way more stable, right? These are a little bit more transient interactions, and so for these guys, you can like sonicate them. You can do quite a lot to the prep, and they'll still stay pretty stably bound. And so you just need to be careful of this. There's always lysate mixing experiments that you can kind of do to test this. But I'm going to show you that in some cases, it's unlikely that this actually matters a lot. So this is the data set where a technique was used to map the actual binding sites, right, with cross-linking and digestion versus a RIP. And basically, you see that there's a lot of transcripts that are found in this clip, and much less in the RIP. So if you look at the number of binding sites per RNA, these have very few. They only have a couple. If we look at the sort of overlapping group that are found in both, these tend to have many binding sites per mRNA and are therefore likely to be like a more stable interaction, or at least probabilistically more stable, right? There's more possibilities at any given time that at least one protein is bound to the RNA. But then this sort of like 485 out here that were not detected in the cross-linking. So this protein here, it stabilizes RNA. So when you knock it down, you should expect to see its targets go down so that they would be lower after knockdown. And so if we look at like genes that aren't targets, right, mRNAs that are not targets, this is their sort of cumulative fraction. And targets are all shifted to the right. That means that they're more likely to go down after knockdown, as we expect. But this blue sort of RIP chip only set, right, has a similar behavior as, it's actually a little bit closer to this 1300 than the parclip only. But the point is that they are just as likely to be stabilized by the protein. And so these are therefore unlikely to be just like reassortment, like that these are not like kind of physiologically relevant RNAs, but just RNAs that have good sites in them and, reassort. But anyway, this is an important thing to consider, and you don't always know until you do the experiments. So I have one more. What time is it? Do I have? Oh, it's 2. Okay.
It's a little bit after, but so folks that have to drop can drop. But if you wanted to talk a few minutes longer, Neil, you're welcome to.
I just have one more sort of slide that kind of speculates on, if we think about binding sites and how that relates to the whole RNA, enrichment of the whole RNA. I think this is kind of important because if you're doing a RIP, you're getting the whole RNA. So when we do this for this RNA binding protein QR, the more binding sites that were determined, the more enrichment there was in the RIP. So there was like a relationship where more binding sites equals more overall binding. But for FMR1, this was not the case. So the x-axis is the enrichment in the RIP, and basically all these lines overlap, and they're color-coded by the number of binding sites. And it just doesn't matter. You're not seeing clear enrichment. However, when you separate the sites out into sort of whether they're more in the coding sequence or the 3' UTR, now you start seeing like different behavior. And I guess like the point I'm just trying to make is that different proteins behave differently. And I mean, what it comes down to is having both of these data allow you to really get a better understanding of what's happening. But this enrichment is oftentimes a really valuable thing to consider, even in the absence of knowing, what the binding sites are. So I'll leave you at that. So let me just say thank you to folks in the lab. Thanks for the invite. And then like a lot of this is from my PhD and early postdocs. So it's pretty fun to go back through a lot of this stuff. And you've asked questions, but if you have any more, I'm happy to stick around for a little bit.
Thanks, Neil. Give it a little applause virtually.
Thanks, Neil.
Yeah, welcome.
Any questions?
Will I be able to get a recording of this? Ashley
Yeah.
had another meeting at the same time, so it would be great. You said a lot of things that will be helpful for her.
Yeah, this should all be recorded and will be posted on the Cancer Center's YouTube.
Perfect. Okay, thanks.
Great. All
Okay.
right. Well,
There's nothing
thanks
else.
everyone.
Thanks, Neil. Appreciate it.
Yeah, no problem.
Okay.
Take care.
Bye, everybody.